Okay, so we tried to have a mix of panels and presentations. Here we're going to have a series of presentations on use-inspired research. So then this will really highlight this sort of research for addressing soil health needs in desert agriculture. So the first question is, what exactly does use-inspired research mean? I talked a little bit about this before. I think the key is that it means that you're going beyond basic research, which is just about gaining knowledge for knowledge's sake, which still has value, right? We're not putting a value judgment on it. It still can be important down the road. But instead, use-inspired research refers to science that leads to foreseeable benefits to society. Use-inspired research does not need to have immediate applications, but it does need to have a rationale, conceptualization, and a research direction directly driven by potential use to which the knowledge will be put. So we're going to begin. In each of these presentations, we ask to be about 15 minutes, and then there should be time for questions. We're about 10 minutes behind schedule, which might cut into lunch a bit, but we want to make sure that there's time for these presentations and questions that follow. We're going to begin with John Chorover, who is the Interim Associate Dean for Research in the College of Ag, Life, and Environmental Sciences. So I'll pass it off to you.
So it's a pleasure to be here today. Thanks, everyone, for being here as well. And many thanks to Joey and Stephanie and Sonnet for organizing today's session. I'm here representing sort of research in the college as an interim associate dean for research. And let me see. Oh, look, it went back and I'm pointing the wrong direction. So, but I also, am very close in my heart to soils. Not as close as I will be eventually, but very close to soils in terms of my own background. I'm a soil chemist by training, and so in the Department of Environmental Science at the university. So we have actually a pretty long history of soil science research at the University of Arizona. Dates back to the foundings of the university. In 1973, there was a merger between the agricultural chemistry department in the college and the Department of Agricultural Engineering that formed this department that had soils in its name sort of for the first time. In 1985, agricultural engineering split off from that unit and it became a Department of Soil Water Science. And then in 1988, there was this environmental research lab down at the Tucson airport that was conducting research on a variety of environmental issues that joined the faculty in that department, and we became this department that many of you may have been familiar with for a long time, Department of Soil, Water, and Environmental Science, or SWES. And then in 2020, the name was changed to Department of Environmental Science to simplify the name and make it more comparable to the name of our majors, so students could find us easily. But always that department and other departments that I'll talk about in a little bit in the college have maintained a very strong focus on soil systems, and that's still the case in this Department of Environmental Science. That's Joey's home department, Debunker's home department as well, Luisa's home department, who you'll hear from later. When we talk about soil science, what is it? There's actually a professional organization in the United States that's called the Soil Science Society of America, or the SSSA for short. And this Soil Science Society of America has been around for a long time, and it has various different subdisciplines, soil physics, soil chemistry, soil biology and biochemistry, pedology, soils and environmental quality, and soil mineralogy. And there are people who train in one or more of these areas and specialize. What's unique about soil health is that it really brings together in a holistic way, as we've been talking a little bit about earlier, all these various subdisciplines of soil science. And in many ways, I sort of think of soil fertility as being kind of the founding discipline or subdiscipline that has led to the evolution of soil health as a subdiscipline as well. And in that Department of Environmental Science, we have actually quite a few people that work in each of these disciplines. And so the folks that you're seeing here are just a subset of a larger group, not just in this department. That's this one department, but there's others, as I'll mention, coming up. But we have folks that sort of work in all of these different disciplines or subdisciplines of soil science, some of whom are represented here today. So the exciting thing, to me, one of the exciting things about this sort of field of soil health is that it's advancing quickly. It's sort of a cutting-edge area of research that has direct user-inspired applications, but it's advancing quickly in parallel with human health aspects as well that have some similarities to what soil health represents. So in our bodies, we actually have an open system that extends from our mouth to our anus. And what that is, it's a place where things from the environment go in and then they come out the other end. That's referred to commonly as the gut microbiome or the gut environment, within which there is a microbiome that composes much of the activity of what goes on in the gut and has a huge influence on human health. And there's this interface between the organism, us, and that gut microbiome that's made up of these epithelial cells where there's communication across that boundary. And in many ways, I think of that very similarly to, or analogous to, what we see in soils, where we have plant roots that extend down into a soil that is enriched in a microbiome, where there's communication between the roots and the microbes through the transmission of different types of signaling molecules. And there's a system that's more holistic than the organism itself, the plant, that gives rise to the health and productivity of the plant, and also the health and productivity of the microbes. So I think of these as being very similar types of analogs in science. And in order to be able to understand how to work in those systems, and we've talked about this already, one needs to be able to develop an approach and technologies associated with first resolving the microbiome that exists in soils, just as it does in the human gut. And we have several faculty that work in that area in the College of Agriculture, Life, and Environmental Sciences. For example, Albert Barbaran has been working on developing a workflow for resolving the soil microbiome that involves going out to the field, collecting soil samples, extracting the DNA, conducting high-throughput sequencing, looking at either phylogenetic, who's there, or functional gene composition. We had some conversation earlier about the functionality of microbes. One can characterize the functionality of microbes based on the genomics in the soil sample, inputting that into bioinformatics software, data analysis, and getting some information about who's there and what they're doing. That's a critical component of understanding soil health. It's something that was considered to be sort of exciting in this earlier panel conversation that we had. We have other folks in the College of Agriculture, Life, and Environmental Sciences that are doing research on what the microbes do in terms of producing molecules, signaling molecules that transmit information between microbial communities or among microbial communities or between microbes and plant roots. And one example is Professor Malak Tufeli, who does a lot of work using high-resolution mass spectrometry to characterize what are the small molecules floating around in those pores that are having an impact on how the plants are growing, how the microbes are influencing plant growth, and things of that sort. We refer to those small biomolecules as the metabolome. What are the metabolomics of these microorganisms in the soil that are affecting plant growth? They have influences on carbon, phosphorus, nitrogen cycling, and they have large influences on the way these systems respond to change. Also, some of the folks that have been doing extraction of DNA have taught us that, wow, there's a lot more microbes, strains of microbes in soil than we can actually work with in the lab. We can't seem to grow them in the laboratory. Like 90% of them can't be effectively grown in the lab. So we need people who are focused on growing those microbes in a high throughput fashion, developing better techniques for growing the microorganisms. And in the college, we have some folks working on that. Paul Carini is one. This can help to answer those questions of, how can we learn from what these microorganisms do in the laboratory to affect things like soil fertility or crop health? How do these microbes influence those types of characteristics of the field plants? And the only way we can do that is by learning how to grow them and manipulating them and putting them into the systems of study. So that's another area where some of our, whoops, I might have hit the wrong button. That is the wrong button. And of course, at the sort of center of all of this subdisciplines in soil science is this issue that we're talking about today of soil health. And we have several folks in the college that are working in that area, all of whom I think are here today, and you're going to hear from as well. And what these folks are working on are ways, I think, to address some of the questions that were raised in the panel session. What are the appropriate types of metrics that one can measure quantitatively to give an idea of what soil health means? What does soil health mean in an arid system? And how do we measure it? What are the links between the amount of organic matter we have in the soil and our capacity to grow plants or store water in the soil? What are some types of amendments or biotechnologies that can be applied to these types of soil systems to enhance soil health? Once we know something about how to measure it, how do we make it better? How do we get higher on the scale? Cover crops is one example that's been discussed, but there are others. Amendments with different types of organic matter, biochar, compost, amendments with microbial consortia, these types of things. And of course, an understanding of soil health extends well beyond our capacity to handle it within the soil science discipline itself, right? In that sense, it's a more holistic field of inquiry than just soil science itself. We need to work together with plant scientists, and so fortunately, we have an outstanding School of Plant Sciences in the College of Agricultural Life and Environmental Science, and many of the plant scientists are sitting at a table over there. I see them, and will be presenting today as well. And of course, why do we care about plants when we're really concerned about soil health? I guess your question would be, why do we care about soil health when we're really concerned about plants? But the reason we care about plants, of course, is because plants provide the feed source, the organic matter, the microbial food sources that are released into the soil through root exudation of organics, root senescence and turnover. That fuels the microbial cycling of that carbon. That carbon gets absorbed onto the surfaces of minerals, starts to form aggregates that give rise to the physical properties that those soils need in order to support plant growth. So the interaction and the effects of plants is critically important. And then finally, we do need to be thinking about how fluids move through these soils, particularly water. What's the behavior of water in the unsaturated zone, which sits above the water table, where most of the plant growth is occurring? And again, at the University of Arizona, we're fortunate to have an outstanding group of scientists working in water science. We have in the College of Science the Department of Hydrology and Atmospheric Sciences, which is one of the best departments in the area of hydrology. We also have hydrologists that work in our college as well, but we work very effectively across college boundaries at the university. So we share an interest in dry lands agriculture with a large portion of the globe. We're not alone. 41% of the Earth's land surface is made up of dry lands. that range from hyperarid to dry subhumid. And so what we do here and what we learn here has great application elsewhere. These are also some of the fastest growing areas in terms of population. Drylands in the United States and globally are the fastest growing population centers on the globe. So these are areas that require our attention. They also are the most productive in terms of arid agricultural lands globally. And there's more of them every day. They're expanding under climate change. There's been a 7% increase in what's defined hydroclimatically as drylands since 1948. So what we do here has direct application elsewhere. Of course, we're all familiar with graphs like this of data in the lower basin that show increasing basin temperatures and decreasing Colorado River flows, so decreasing availability of water, increasing sublimation, increasing evapotranspiration. So even though there's not a big decrease in precipitation in the Rocky Mountains, there is a decrease in the amount that's coming out and delivered into the Colorado River. And so we need to be able to work with smaller amounts of water, which is something that we're talking about here as potentially soil health being a way that can help us with that. We're going to hear more about this since I see I'm run out of time just now. We're going to hear more about this report from Kim Patten, I think, in the next presentation. And so I'm going to also, we will hear shortly from Duke Pauli about a center that's been just recently created in our Division of Agriculture, Life, Veterinary Sciences, and Cooperative Extension at the University of Arizona that I think directly addresses some of the plant science contributions that interface with soil quality issues that we've been talking about. I just want to leave you with this final slide to sort of highlight that at the University of Arizona, we have the capacity and we support the kind of research that's being discussed here, and we can really work across multiple scales, from molecular scale using state-of-the-art techniques for understanding molecules and genes and genomes in soils, all the way up to landscape scale processes and a variety of different types of sort of agricultural practices, including field scale, ranching types of activities in addition to intensively managed agricultural fields like we have here in Yuma. So thank you. I'm happy to take any questions or comments at this point.
Question on for the use in agriculture they were talking about the 21 days before harvest that we use different water treatments. We use I'm an irrigator, so I used chlorine and I used organic peroxide, and that is add to the water to sanitize the produce, right? And all my understanding with the soil is that it's a living organism, and it's kind of like us adding these water cleaners as it's affecting one way or another the soil, right? Yeah. And my question is it actually something measurable that the effect that it's doing the good? And we're using this to eliminate E. coli and all the bad organism, right? So we're treating with the peroxide and chlorine the bad organism, but at the same time it's affecting the good microbiome. Yeah, the good organisms. So do you think is actually we still using is it worth it to keep using this, or what do you think? What's your?
I think that's a great question. I mean, I think that there's now a requirement that the water be sanitized. So the choice comes down to what sanitizer makes the most sense and what approach do you want to use. And maybe UV light irradiation would be less impactful. But no doubt, when you introduce an oxidant into the water, You'll kill some of the pathogenic organisms in that water, but to the extent that the oxidant remains in excess when it's introduced to the soil, it will kill beneficial microorganisms. It will oxidize organic matter. And so there's no question that it's not specific enough, essentially, to just go after the bad guys. It goes for the good guys, too. And I thought that the mention that Matt had earlier in the panel, that it had an impact on spinach yields, was a very important, significant result as well. Thank you.
All right, and on that, because you've heard about it a couple times, if I run into any issues with questions, I'm going to call on Luisa, who just walked into the room because she was part of this commission, and Paul Brierley, who I don't see at the moment. So Kim Patten, I serve in the Central Research Office of the University of Arizona. I think I have the best job on campus because I get to learn from all of the different faculty what their research interests are and then try to align that with a lot of federal funding opportunities. So I believe that was why I was asked to be part of this commission, which was established in December of 2022. And we were asked to really think about the concrete steps that the University of Arizona, and I want to highlight that this was really focused internally at this point, that we could take to help position the state of Arizona as that global leader in climate resilient agriculture and food production. And I want to highlight that that was actually, it was Laura Condon and Joaquin Ruiz who took over for Paul Brierley. And, but there were a number of other people, including Parker Anton, Jim Beiser, Sharon Collins, Levi Escara, Luisa Ikner, who just walked in, Sharon Magdahl, myself, who participated. And then there were over 130 individuals that submitted responses to our survey questions in terms of feedback from the community. Many of you may have participated in that. We used focus group opportunities across the state. We need to do more of that. I will acknowledge that right now. And we worked with some folks from our Social and Behavioral Sciences College to actually go through all of that data that we collected, because those of you that know open forms and actually coding that data and turning it into something useful is really, really tedious work. So I appreciate the help of Jane Zaviska and her team in the College of Social and Behavioral Sciences that helped us out. So we've heard about the number of threats that we were addressing with this report, including the fact that, as John just mentioned, the world is growing drier and hotter, and we need to be able to continue to produce food for the growing number of individuals that are calling this world home. So how do we do that? We also talked quite a bit about the socioeconomic and cultural concerns, especially around policy that might drive to loss of agricultural land, the fact that we need a workforce for this work that we're doing. And I actually should take a moment to say thank you to the people in this room, because I am one of those converts who really didn't think about where my food came from until I did an ag tour and a farm tour out here, and I have drank the very salty water, and I am sold. This is such a tremendous area and unique location for the workforce, for the opportunities that are here, for the food that you produce for the nation. So thank you all for what you're doing in this room to make that happen. And then we also talked quite a bit to impediments to adaptation, including our own siloed resources on the campus of the University of Arizona. And then we were thinking about how we really need to be thinking about this as a systems process rather than an individual research process. So we've heard quite a bit about why the University of Arizona, we're the state's land-grant institution, and we are responsible for the sustainable future of this state. We have extension offices in each of our counties, including on and with 5 Native nations. We have that expertise in water, agriculture, and food production across multiple colleges. And the list goes on, but the one that I really want to highlight is one that is not on this slide, but that is that our environment is really an advantage, right? Again, we are in this amazing opportunity and location that can serve as a test bed and a area to produce solutions for the rest of the world. Opportunities for global leadership. We've chatted a little bit about this, and I'll talk more about that at the end, because this is one of the recommendations that we made. In terms of recommendations, we really tried to focus it again on things that the University of Arizona could do, which is research, the education and training piece of that, connections with our community through the extension offices, and then economic development, spurring what can support the state of Arizona in terms of driving this, I believe it's $23 billion enterprise of agriculture in the state. So one of the first recommendations was that we create this Institute for Sustainable Food and Water Agriculture Systems. And this was the overarching institute that would work across the campus, that would actually bring together all the experts and help try to address that systems level challenge in arid land sustainability for agriculture, food, and water systems. We would leverage the existing capacity that we have with partnerships to help secure this and secure funding. And one of the things that we heard earlier was difficulty in finding funding that are multiple years in time scale. And one of the things that we can do to help address that is to do exactly this, which is to bring in and support use and user-inspired research, because that's where you actually find funding for that 10-year scale. And it makes it much more competitive. I'll be totally honest with you about that. Some of these funding opportunities that have that scale have a less than 2% fund rate. But having this type of opportunity to hear and listen from those end users is what will help us address those and actually get to the point where we can obtain federal, state, and local funding for those. So just a few goals of the Institute here. We want to obviously build partnerships. We want to accelerate that use-inspired research. We want to help support policy and co-develop solutions, strengthen our cooperative extension opportunities, develop the agricultural workforce, share that knowledge, and then connect the communities and policymakers. We would bring together the expertise across our colleges, in the university, and then partner with the academic units, centers, and institutes. Sorry, I've got my notes on one screen and this here. You would think I can make this happen. I also can't garden, so just so that you all know, my only apocalyptic skill is being able to drive a manual transmission, so. If you need me, I'm here for you. 6 feet. I guess I would fail on the large scale transport as well. God, no apocalyptic skills. It's killing me. Okay. The one that you've heard additionally is the Center for Soil Health. This is one that we're obviously very interested in today and are moving forward as you are all here talking about soil health. So that purpose, as you've heard, is to really bring the research capacity to address these major research issues together, to integrate what you all are doing on the producer side and help support this structure, to scale that to a process and system that is actually scalable, right? We heard that earlier as well. And then provide that science to support the policy making and the decision making. We would love to see this live within the Division of Agriculture, Life, Veterinary Sciences, and Cooperative Extension, which includes the College of Ag, Life, Environmental Sciences, the Arizona Experiment Station, and Cooperative Extension. We would love to bring in faculty from engineering. I think we heard some interest in AI-driven decision-making and data use earlier from one of our members from engineering, the public health school, and many others from the university. We feel that this would be a fairly modest way of providing institutional support to help move this whole process forward in a very meaningful way. And we anticipate that we would be able to obtain federal funds specifically and then foundation funds as well. And if anyone's interested in a potential naming opportunity for the Soil Health Center, go talk to Alex. The next situation that we recommended was the technology and innovation hubs at Maricopa, Yuma campus ag centers and up at Biosphere 2. For those that haven't been to Biosphere 2, it is a phenomenal opportunity to do research at scale that is not possible in a lab. But we felt that these areas, and particularly Maricopa and Yuma and campus ag centers, could be promoted further and could benefit from further investment in these technology and innovation hubs so that we could do additional work at the scale that's meaningful, that can then produce the answers to questions that you all have as the growers and producers. So here is exactly what I just said in very long slide form. The structure of that is to serve as the collaborative spaces for faculty, producers, and industry. I was out here last month and hearing about the phenomenal opportunities that are actually taking place at the Yuma Ag Center and the potential to work with industry to have additional lab space, to answer the questions that we've been talking about today and in other forms and forums. Again, we think this would be a less modest investment, but a long-term strategy that would result and benefit the state of Arizona. One of the final recommendations that we had is really expanding partnerships, and it's expanding partnerships with our tribal ag partners, but also globally. So 4 is expanding partnerships with tribal agriculture. And what we really wanted to do was spend a bit more time within our indigenous partner communities and learning from those practices and advancing farming methods. I was recently in Washington, D.C. talking to the director of the tribal office for the USDA, who was really talking about how we have some phenomenal researchers at the University of Arizona that are very clearly linked with our tribal communities and how we can learn from them, and particularly around dry land and arid land agriculture. And so I would love to see this piece of this recommendation move forward. For those that are not familiar, as part of our extension office, we also have the federally recognized tribal extension program for TEP that could also be incorporated into the structure of listening and learning. And then the final recommendation was to establish new and strengthen existing collaborations with institutions in arid and semi-arid regions around the world. And for this one, we already have very strong relationships with France. I was talking to Dennis Donahue this morning about the fact that the FIRA conference up in Salinas is actually from the French ag tech folks, FIRA. And so we work with the CNRS, which is a science funding agency within France, very closely. In fact, we have the France-Arizona Institute for Global Grand Challenges at the University of Arizona, so we could continue that relationship. In October, we hosted the UK consulate on the campus of the University of Arizona, who was very interested in talking to us about a number of things, including agriculture. And I believe, Stephanie, you were also involved in that visit. We also have existing relationships with Australia, particularly Western Australia, and Oman. And so really thinking through how we can partner and strengthen those relationships abroad to help really make Arizona and our institution home for thinking through arid and semi-arid agriculture. So on that note, because I am down to 46 seconds, I did want to take just a moment to state that we've moved from the Presidential Advisory Commission into a Presidential Implementation Commission. That commission has expanded. There were 9 of us on the advisory commission to start. We have about 30 participants now on the implementation team, and that includes additional faculty whose research we highlighted but did not necessarily represent. We brought Stephanie on board for that implementation committee. We have some folks from the Southwest Center for Food Sciences, so from the Social and Behavioral School, and I have 4 seconds left, so we are good. Any questions?
Thanks, Kim. So all this cooperation, but what about ASU or UC Riverside?
That is a great question. We actually, one of the projects that we recently applied for, made it into the semifinalist position, but did not receive a finalist for, was what's called the Regional Innovation Engine project. And I actually think this would be a really cool opportunity to take back to that program when it reopens in April. Hint, hint to any of the researchers that are listening. But it's a $160 million regional innovation opportunity over the course of 10 years. And in the previous round, we partnered with Davis, but also had conversations with Riverside. And so we're very interested in working with members of the UC system, the Cal State system, SESU, Imperial Valley College. ASU, we love working with them faculty to faculty. Happy to have that conversation.
I wanted to ask about the status of these new institutes.
Yes.
For example, the Institute for Sustainable Food, Water and Agriculture System. When should we expect them to start?
Well, so their recommendations right now, and part of the work that the implementation committee is taking on, is to actually determine how best to implement and to make those a reality. So I would argue that, and we did this intentionally, that the Center for Soil Health will likely occur before the Institute.
You told me not to ask a question, but I'm going to anyway. So we've heard a number of fantastic researchers that are involved in some cooperative extension, but you'll see it's outweighed. There's a ton more researchers that are working in this area, and there's 3 or 4 of us, right, that are covering the state related to implementation, boots on the ground, translational science. And so questions for some of those objectives about hiring, right? So is there space in some of those objectives to not just pull folks together that are existing, but to hire additional folks that can help share that workload for cooperative extension?
In my ideal world, and I believe that of the commission, absolutely. Absolutely. And so I think you saw that one of the recommendations was actually to oh my goodness, hire, not fire, hire 4 faculty lines. And I believe that was part of our recommendation, was to actually place those and ensure that extension was recognized as part of that. Because as you said, boots on the ground, especially on this type of cooperative work. And when we're thinking about partnering with UC Riverside, Imperial Valley Community College, it is the boots on the ground that have those relationships and ensure that when we go to them as the research office, right, they know that we're there with a reasonably authentic desire to really partner and demonstrate. And frankly, for the researchers that are in here, that is my goal. Whenever I'm talking partnerships, I really want to be able to support and ensure that your partnerships are really elevated. And so it's about you and your work, not.
All right, thank you very much, Kim.
Can I just make a really quick comment? I just want to say, Kim, your office and Kim's group is a very big supporter of Yuma Ag and the work that YCEDA is doing, so I want to thank her for that.
Good morning, everybody. It's a pleasure to be here and to actually share some of the work that we're doing within the college and how it addresses some of the challenges that our state and nation are facing. And with that being said, recently, you've already seen it mentioned twice, a new center was created called Agro-Ecosystem Research in the Desert, makes a nice acronym, Arid. And so this center was really created to address fundamental problems facing arid land agriculture. So what specifically is the Arid Center? So it is a new center based in Cal's, housed in the School of Plant Sciences. It was basically, fundamentally, initiated to identify and solve challenges facing arid land agriculture. As we all know, we've heard about, John, that was an excellent introduction. In our state, nationally, globally, aridification of our farmland is becoming a huge problem. And given that agriculture supports directly or indirectly 23% of the global population's income, the challenges and solutions that we're facing, translate across the world. And so what became apparent is that we needed more of a systems approach to this challenge. And specifically, asking the question, how do we feed our planet without destroying it in the face of changing climate? When I arrived here 5 years ago, I'm mainly just a plant geneticist and breeding, but after 5 years of growing crops in our unique environment, it became pretty clear that it's more than just plant genetics that are going to get us there. And so that's really what implemented and was the impetus for forming a center to basically synthesize and bring together these different resources to form a partnership basically across the college to address these fundamental questions. That being said, the vision of the center is to advance environmentally sustainable agricultural practices through the understanding of complex physical and biological interactions with air-land ecosystem as well as humanity. One of the things I'll highlight next is that as this problem occurs across our state, nation, and the world, it has a direct impact on our rural communities. And so finally, to achieve this goal, it's been largely a focus on synthesizing our different expertises within the college, specifically technology, our expertise in basic science research, data science. UA is one of the leaders, I'd say, in the U.S. In terms of data science literacy. So bringing all those skills and expertise to bear on this problem. But why is Arid actually needed? Did UA already have something that could fit this need? And when we looked around the landscape, the answer was no. I mean, there are other institutes that have some overlap, but nothing really that addressed the nexus of agriculture, arid lands, and humanity. And so we already know the challenges have been listed by John and Kim, that we have changing weather patterns, more frequent extreme weather events. Who remembers the heat wave last summer? Unprecedented, right? We're feeling the effects, and we're running out of time to come up with solutions. We know that we have decreasing access to fresh water. Drive through the Stanfield Irrigation District, and you'll see the impacts already. Additionally, there's other costs, so increasing crop inputs. For all the producers in the room, I would argue that things probably haven't gotten cheaper for you. We think about access to labor, another critical issue. And so we have emerging crops and pests, so we have kind of this perfect storm of events that are happening right now in our state. And Arizona really is at the forefront because we are addressing these challenges before other states are facing them. When I got here a few years ago, I had to think of a way to brand my research, and it was basically Arizona, the climate of tomorrow, today. And I would stand by that. The temperatures that we experience, the lack of water that we have, these are the challenges that the Midwest will face in 10 to 15 years. And because of that, it gives us a really prime example of how to come up with these solutions, implement them, and be successful, and be a paradigm to the rest of the country. So one important thing when forming this center was talking with Paul and basically being like, Paul, I don't want overlap or competition. And so looking at YCEDA, just some fundamental bullet points, it's applied solutions to grower challenges. And I would argue this is what makes YCEDA so successful across the nation when you look at other programs, is that the stakeholders are actively engaged in the program. Because of this public-private partnership, I would argue that under Paul and Stephanie now that, again, the impact that's being had in, our agricultural sector is huge. Obviously, YCEDA is focused on the crops that are grown in Yuma. Obviously leafy greens, durum, wheat, some tree nuts, date palms. So it's kind of regionally specific. And then, but again, to focus this on solutions-driven research. At the end of the day, the growers want an answer that they can use. For Arid, we are kind of at the opposite end of the spectrum, by design. We're a basic science research organization, basically out there for knowledge discovery. We're publicly funded, and we're focused on crops that grow throughout Arizona as well as the nation, so cotton, sorghum, we do lettuce, we've worked on maize, tepary bean, a whole host of things, but it's really all about exploratory research. And the reason it's about exploratory research is because it really completes a perfect partnership, if you will. So again, YCEDA is very grower-driven, focusing on the challenges and coming up with the solutions that are needed here locally. That's fantastic. That's something that Arid can't replicate. But what we can do is drive research, basic science research to identify those problems that maybe haven't crept up in your fields yet. Start doing the fundamental research that costs money that maybe you don't want to fund. and do more of an academic exercise, if you will, as my advisor used to say. But basically, the complementation of these two organizations represent an entire pipeline from the basic science to applied solutions that can be delivered in your field. I would argue that by having a complete pipeline, we can do this fundamental work, we can de-risk it. As growers, you have to, make the most of your money, and so if we can assume the risk at the basic science side, come to YCEDA with more of a finalized project that's worthy of your investment, you have the opportunity to look at the data, to vet it, and make an informed decision if this is something that growers should support. So I just wanted to talk about one. I could stand up here all day and tell you about Arid and what we're doing, how great we are, but I guess I kind of want to highlight one case study that might resonate with you locally, and that's looking at the lettuce microbiome. If you read any trade magazine right now, progressive farming, successful farming, forage and crop, everything is about biologicals and the carbon market. Carbon market, I think it's still kind of snake oil. Biologicals, I think there's more research to support that. And so that's kind of what I'd like to highlight today. And so this work is being done in collaboration with Betsy Arnold. She also happens to be the associate director of Arid, and this is largely one of the reasons why a lot of this stuff is getting off the ground is we had this fundamental question of, can the lettuce microbiome actually improve plant performance? So we know that genetics, when you breed plants and whatnot, obviously control a lot of what you see in the field and a lot of what you harvest and yield. But we also know that traditional plant breeding is pretty slow. A decade to go from a cross to a really released, improved cultivar. That's too long. We're already seeing the changes in our environment now. So how can we kind of circumnavigate that and speed up the process? And that's what kind of led to these fundamental questions of, we know the plants are interacting with the soil environment. We know the soil is diverse. There's a lot of stuff happening there that we don't know about. But what extent does this interaction actually happen? What is the crosstalk between the microbes and the plants themselves? And then, how does this root environment, how does this actually impact the above ground performance? Does this lead to increased yield, better drought tolerance, shelf life, leaf properties? And finally, how does a leaf microbiome impact micronutrient availability? Lettuce is the #1 consumed fresh vegetable. So, if you can improve the nutritional quality, you've just added value to the product. Consumers now, that's we see that more and more in the marketplace with, people eating fresh fruits, vegetables, thinking about blueberries, raspberries, things like that. So it's value added to the crop that wouldn't cost much. And then finally, how can we translate this work to something YCEDA might be interested in? So the work I'll talk about today is in conjunction with UC Davis, the USDA, and Salinas. And basically, this is part of a trial that I run in Maricopa. We are growing a panel of 250 different lettuce varieties, representing all the genetic diversity and phenotypic diversity in cultivated lettuce. For this specific work, we looked at 12 varieties representing 6 different market classes. For those of you familiar, they're at the bottom of the screen, if you can find your favorite. And so to do this, basically, Betsy's team came up and had a high throughput way to collect these samples. So basically, we're digging up the plants, we're sterilizing the root tissue, we're getting it macerated, being able to get that into culture, and to do the sequencing, same thing with the leaf tissue. And we're looking at the bacteria and fungi that are naturally living with the plants. As mentioned, I run the UA Field Scanner. This is the world's largest outdoor phenotyping platform. So we're collecting a constant amount of data on how these plants are growing and responding to the environment. We're also doing a lot of root phenotyping in conjunction with Dr. Alex Bucksch, who's here, and then micronutrient profiling of the leaves themselves. So we're trying to look at all these different processes to see how the microbiome might impact this. Now this graph, before you just glaze over, it's pretty easy to understand. So all these colored dots, if the same colored dots are closer to each other, that means the microbiome of those samples are more alike. So if we look at this blue corner, oh, it died. So the closer they are, that means the more repeatable, the more consistent the microbiome associated with that plant is. And so this shows us that these different varieties are selectively recruiting organisms from the soil environment into their root tissue. Why, we don't specifically know yet, but we've seen other work with sorghum, cotton, and tepary bean that typically under drought stress, plants actively recruit microbiomes that help them in terms of stress resiliency as well as their growth. So right now, this is some very exciting data for us because it shows that, again, just looking at 12 varieties, there's differences that we can probably leverage. Looking at the bacterial communities within the leaves, similar pattern. We see that among these different varieties, the plants are actively selecting for certain bacteria. Why they're doing this, we're not entirely sure. But our next question was, well, okay, if we look at foliar chemistry, how does this track? I apologize for the graphs. I know they're not great, but basically what we want to point out is under zinc, iron, and magnesium, and the 12 genotypes that we're assessing, we see variation among those genotypes. It's very specific. There's marked differences between just about every one of them. And so to tie this all together, basically, is we can put this together to assess bacterial richness in leaves actually tracks with the nutrient content. And so what this suggests is that through modification of the microbiome within the leaves, you could actually change or improve the nutrient content of the lettuce. To just kind of wrap this up, if we look at the lettuce microbiome, we know that the lettuce is actively recruiting different fungal and bacterial species. In case of the leaves, it's tracking with the nutrient content. So because of this, and we see this very distinct variation, it suggests that there's some type of crosstalk between the plants and the bacteria that likely has a genetic underpinning that we can leverage for the advantage of lettuce production. When we're looking at connection to agronomics, the work presently, we're still analyzing the samples. This is all from last year. We have another field trial that's going on right now in Maricopa, repeating everything to make sure that it's just not snake oil as well. But basically what we're looking at is, can we associate the microbiome with these other traits such as hydration status, shelf life, and yield? So, does this microbiome, is it a way to be profitable, increase the plant resiliency, increase yield? And then in terms of future work, if these variations hold up with the microbiome and performance, how do we get that into your field? So if we talk about like seed pelleting, it's a simple way to take a combination of beneficial microbes, put them in solution, pellet the seed that's already commercially done. And so now this gentleman asked a question about you're putting bleach on your fields and you're basically killing the natural microbiome. This offers us a chance to get those microbes back on the plants where they're actually needed. And so when we get to this stage, this is why it's imperative to have something like YCEDA to interact with and work with. So at the front end, we can do all the basic science research, but hopefully towards the halfway through the project, we're now coming to you as stakeholders and saying, A, is this of value? And B, if it is, how do we get to the next step so it's actually beneficial for you? And so this is just kind of one example I wanted to highlight in terms of the work that Arid's doing. We're actually taking this, the center is doing a lot of work in cotton as well, sorghum. I know those crops aren't specifically of interest to Yuma, but it's this kind of same paradigm of how do we take a systems level approach to improve plant resiliency in a meaningful manner. So again, we know that agricultural production in the U.S. and Arizona and the world is under immense pressure from our changing environment, the constraints of water. And so really, thinking about how we can leverage our strengths here at UA to address those problems. And I think Kim was right. We had this really unique opportunity to be leaders in this space. And this was really one of the reasons why Arid was formed, was to basically capitalize on the resources that we have, the expertise that we have to make meaningful impact to actually developing solutions. Most of the time I can speak with plant breeding, you come up with a improved variety, you put it out into a field trial, it doesn't do that great, right? They're regionally specific. There's a lot of complexity happening. And that's why having a systems approach to these types of problems can be so beneficial. And then finally, just in terms of a partnership with YCEDA, it forms a nice pipeline that we can do the basic science to get the primary work done and then come to the stakeholders with something that they can have confidence in terms of a higher return on investment. So that everything's kind of been worked out, and by the time you're testing it in your field, the chances of success are much higher. With that, I would like to thank you for your time. I guess if there's any questions, I'd be happy to take those.
Hello, Robert Bassan, Yuma County Cooperative Extension. I have a question. I see a lot of research being done to improve the nutritional content of leafy green leaves. I'm curious, where that initiative came from, if that came from industry coming to you asking for that, or if you're just assessing the needs of the consumers directly, and whether there would be an increase in profitability for the companies if they did have some products that had higher nutritional content.
Sure. So luckily for me, Robert, working on that SCRI grant we have with Richard Mitchell-Moore at UC Davis, it was kind of born out of a conversation with him. He's obviously much more tied into the lettuce community than I am, but basically kind of highlight like as one of the fundamental things, like if it's the most consumed vegetable, it offers the best way to improve consumer health. And so I think for him, it was a conversation of industry going to him and bringing it up. I'm coming by the information secondhand, full transparency. So I think that's kind of what was driving it. But he made the point that it seems like consumers may pay for it. If you look at what happened with like pomegranates, right? All of a sudden there was a pomegranate craze because everybody said palm was like great for you. Whether or not that's true doesn't matter. If you look at that little juice cup thing, that company's gone through the roof in terms of their profitability.
Now this might be a stupid question, but I'm just curious if there's any association with the microbiome products you're using, and an association or interaction with the foodborne pathogens that we're preventing in the food grains.
Excellent question. I apologize for not mentioning that. So the leaf samples that we were isolating the bacteria from, or Betsy did, so these are non-pathogenic. These are all beneficial microbes that occur naturally. So for your specific question, I'm not sure there's any, if we can say whether or not like the beneficials are outcompeting. I don't know that. But thank you for the question. That was excellent.
All right. Hello. So yeah, thanks for the introduction. Great to be here, and I'm just about a year here. So a lot of this is not done on lettuce. But yeah, I work on roots, and I'm a phenomicist more or less, right? So originally mathematician and computer scientist, but I fell in love with plants 20 years ago and started making a lot of data on plants in ways that we normally don't think about. So let's make the problem first a little bigger to give you an idea how we can think about roots in the soil. And that is, if you think about the three big carbon sinks that we have in the world, it's forest, it's the soil, and it's the ocean. And they're all getting destroyed, right? We harvest the forests right now. Deserts are expanding, you know that. And the oceans come over, so it becomes a lot of salinity. And we call that just degraded land. That's what it is. And the question we have, it's very easy. Can there be something green on it? I think we're on the same page with that. Okay. The way we think about it is very simple. It's not about every single nutrient. It's about, hey, in good soil, resources are very close to the plant from a plant perspective. And then if you go into degraded lands, it's typically further away. So we are just more distant from the plant where these resources should be. Very simple problem that applies to a lot of things. Now, what you learn is this. There's something missing in your soil, and your root and your plant react in a certain way. If you look here, there's, for example, let's take drought conditions, right? Your root gets very small. So that's what the plant does. Just intentionally no citation there, because that's a textbook that is used also at U of A. And if you go one step further and say, let's check if this is right, and I'll show you what is in the soil in a whole field in Wilcox. Yep, then I see this variation. Huh? Strange. Any idea why there's so much variation? It's not one response like they told us in the textbook, right? Well, the problem is pretty simple, and now I'm taking my math hat for a second also. I love the plants. But if you look at the last 100 years of field ag research, we are completely undersampled. We're looking only at the things that are most common in it. It's something to also look around you. So what people learned all the time, it's just a fraction of what is out there. And that's where we draw our conclusions from. It's a very simple thing that it's not easy at the beginning. So our idea was we take that all, these were bean roots, and we have a very simple idea. We say, let's see if there are similar ones more often or less often in it. We did that, and for bean, and I just show bean, here in this presentation. There are 5 different kind of root shapes in it, and they have different fractions. And if you look at that, the most dominant one here on the side, so that's data from Wilcox from 2015 and 16. It's about 25-30%. That's where that's the conclusions you draw. That's the amount of data we look in and put into textbooks. That's not what we really wanted. Now, we check that. That happens all over the world in different places, in corn and in bean, common bean. These are really the pinto beans in your burrito. So it's everywhere. And that contradicts a lot of the literature that is out there. Particularly, I just want to point out the middle one, which is about plant breeding, where we still think about this one phenotype, this one root that is in the soil that we bred for. It's not happening. Also, it is not related to the heterogeneity of the soil. We found that out. And also in other theories that you find, they always think about this one phenotype, that one root that is in the soil when they start deducing new knowledge. Well, we thought, what are these things? It's much more important. What are these 5 different kind of roots doing? Yeah, they have different functions. So it turned out, and that was a very nice study that blew our mind at the beginning, some are good in taking things up from very deep, other ones are very good from taking it up from the top shallow soil, and other ones are very good in taking new things up that are just diffusing through the soil, like water, nitrogen, right? Topsoil is typically phosphorus. Deep down you find often water. Nitrogen is often just diffusing through the soil. Okay, let's compare 2 of these beans. One is good in drought, the other one is good at phosphorus. That's what they are officially bred for and where they are coming from. So the DOR is from Chile, the other one is from I think it's from Mexico. Yeah, I think it was from Mexico, the SEQ. So what happens is, if you put them in drought, so we call that 30% volumetric water contact, so 30% of what you normally need. The SEQ goes a little bit down. The other one is not so much affected as you expect. Now, let's put them together. Let's just put one seed next to the other, the different one. What happens? Okay, the drought one is pretty much the same. The other one, jumps up. It's cool, right? Can do that in drought too. Not much happens statistically. Whoa, it gets even higher. Right, so there's something going on where they help each other. So the one that is not so good in acquiring water actually gets the water. If you just take one pinto variety and switch them out next to each other. So the reason for that is really simple. If you look now suddenly at the spectrum of root architectures that we had here, there's one that sticks out that really changes to something that hasn't been there before. So we are reacting to our neighbors, and something is either sending a signal or exchanging a resource, or something is going on that we want to know. We don't know why that is happening. There's something going on, and there's a lot of basically technology hurdles we still have to solve. To find all the answers, to scale that up to many other plants. At the moment, we just have bean and corn as examples where this is happening and where we have certain combinations identified that show these kind of patterns. There are more effects in this plot, but this is the most important one. There's a second thing I wanted to talk about a very old project that I brought here or dug up from older presentations I gave. And that is something we did in Thailand at the beginning of my assistant professorship when I was still in Georgia. And that is, we looked originally on growth stunting in kids in Thailand. Thailand is pretty far developed. It's not a developing country, and still 50% stays growth stunted, which is strange that you don't expect there at all. Now, what we did is we tried to look into the cassava that mostly the poorer population eats, especially during pregnancy, apparently. And that's what we compared. We could take 2 of these cassava varieties, and one, the R5, makes these storage roots that you eat, similar to potato, very late. So it goes on and on with development. And the other one bulks very early, and that's what farmers want to have. Bulk fast, it's not a year, it's just a few months to get these storage roots, and they are almost the same size, nicely distributed, very marketable and industrial usable. Now, what turned out, the drivers of growth stunting, calcium and zinc, right? And late bulking that developed much, much longer in the soil, even though the soil is, this was done in various places, always had the most content. Also happens for magnesium, by the way. So why is that, right? You need both together somehow. You need really both together. So zinc and calcium play forth and back during development of the cassava plant. So that's something to know. You cannot just throw calcium on there to get more calcium to prevent growth stunting. You actually need also to take care of the zinc ratio with the calcium, because zinc will enable your plant to take up more calcium, at least in cassava. I wanted to throw that out because that's really soil management that you have to do to remove health problems out of your population. And that's just one example that worked very well. Okay, I mentioned that we are really technology limited, and I just give you a little view into the toolbox. Our best tool is still the shovel to get all these roots out of the ground. But our toolbox has a little bit more. So one of the things we have is a controlled soil environment. We have lots of sensors in there to monitor how much soil moisture is at a certain depth. So if you want to have more control, we are not only out in the field, we can use these kind of environments to get more control. And we know exactly later on in the data, hey, there's a wet spot that might be an outlier. We know why. That is most important if you look at these complete populations of plants in a field. Another thing we do here right at U of A at the moment is we start looking at the interaction between two roots to get more insight into that. So what is going on between also in the soil? Are there certain microbes like Duke mentioned in the game? So one of the problems we face is you hardly get these two roots that you see on the left bottom out of each other. Probably you hardly see that there are two roots in it. Now, a simple way is that we develop a method to color these two roots to distinguish them first, and that gives us the label to apply deep learning. But it's a very mechanical process that goes up front to generate labels to distinguish two roots when you remove the soil. So that is a project that goes on already since over a year until you get enough contrast. And in the details, it gets very tricky, especially building a box around is tricky. You won't believe that. A box can easily impact the phenotypes or the roots that you actually observe. Another thing we did is developing a 3D scanner that is actually scalable. So if you think of a CT or an MRI, that costs $500,000 or a few more $1 million. And if you want to make thousands and thousands of roots, you cannot afford 100 MRIs or 100 CT scanners. So we built this scanner from Amazon material. It was about $2,000, $2,500. And over the last year, we actually could make it to like about 2.5 kilogram scanner that we can also transport easily. The other one before, you needed a big truck. So question is, what comes out of that? Which detail? That's the detail we can do right now. So that's a corn root. It's very dense. So we show that example because it's really hard to get all these little root branches out here. If you zoom in, we actually can really go into the root. So we solved, which is really a mathematical problem of occlusion that took us about 2.5 years to crack mathematically to deal with the occlusion and get validated results from such roots. So the measurements are good. Now it's about scaling that to the results and possibilities you saw before. So we will get more noise, more variation we can look into, and hopefully find more hints and more explanations about how these roots interact with each other and perhaps even cooperate. All right. Brings me just to the sum up what I want to do the next, also hopefully 10, 15 years here. And this is really getting to the mechanisms how roots cooperate and help each other in a field. I think that's a mechanism that is totally underexplored at the moment, and the possibility is there. There is even exchange of nutrients between plants known. It's known that this happens, we don't know how, and is there a way to influence that to make them better collaborators? It would be nice. So we have less, but we collaborate with our strengths together. Similar like you do in the center or here in YCEDA, right? We just bring our strengths together. And the big bottleneck we have are the non-destructive soil sensors. How do we sense not only what is in the soil, but also what deforms and develops in the soil? That's a very different story. And so there's several projects at the moment where we explore new sensors that go towards these kind of goals to explore this collaboration and specialization game below ground. And then there's one last thing, we do need more multi-scale models. And that's pure math to simulate what we will never be able to measure. We have physical constraints, physics is sometimes in the way, we cannot help it, but that's another area we get really, really busy at. That's not going alone, right? There are a few, 7, 8, about $10 million of funding behind all of that, even though it's a lot of shuffling. Not everything I know I could tell, so there's already a lot of industry cooperation in there, so the corn results I skipped for good reason. But that's a lab here at U of A that is currently working on these topics. And all the collaborators all around the world and the nation. So I just say thank you. If you have any questions, it's your floor now. Thank you.
Hi, thank you. So when you first began talking about the variation in root phenotype, at first I thought you were talking about some kind of plastic response, but you were talking about different responses to different stressors, right? So this crop plant growing under these conditions of stress and that crop plant growing under those conditions of stress, they're going to have different root phenotypes, even though they're actually genetically quite similar.
So, okay, if you go into the details now, so these are clones. They are 12 generations selfed. There's almost no genetic variation between the individual plants. Okay? So we put populations of them into different stresses. That's what we did. But each plant experiences exactly the same stress, and we have this controlled soil environment. So the variation itself is what we took out of each other. So
then you're getting these different phenotypes.
Exactly. And yeah, and they are very specialized functionally. So that is the new thing that we really saw is it's not just variation because of soil patchiness or anything. It's really they have different functions. And it seems that if you specialize, you can affect your neighbor. So the big question is what are the mechanisms? That includes microbes, bacteria, soil physics. There are many, even just soil biochemistry plays a role in how they can help each other next to the next neighbor. It's a very underexplored area at the moment, but we see that arising in many, many species. Actually, in Africa, many tribes use these effects just by word of mouth. They know which varieties they put next to each other.
I was under the impression, and I'm not a root scientist, but I was under the impression that when plants are growing in close proximity with each other, that they're naturally going to separate their roots so that they're reducing competition, but this is where they're actually interacting and communicating?
Could be. So it's a good question. Do we have an exchange of small RNA between the two that one knows what the other one is as a specialist or as a generalist maybe too? We don't know that. We're on the way to find that out. What are the actual mechanisms that underlie this variation? It's definitely not noise. We know that now. So until probably last year, so this is now the 5th or 6th time that I really show that data. Until last year, you probably thought, oh yeah, that's just noise, and we throw it away. And that's what happened over the last 100 years. We threw it away. We threw all of this variation that is very valuable, apparently, just away.
Hi, Alex.
Hello. Hi, Nancy.
I enjoyed your talk very much. Do you have any, a follow-up to what you just said, do you have any evidence that maybe some of that variation might be linked to some of the variation in the microbial communities?
So we have no measurable evidence. That's the technology limitation we have. So, but there is, for example, we do know that the nitrogen gas is very much exchanged between plants through the mycelium. We know that.
Let's
So, but
talk that much.
maybe, yeah, we can think about these hypotheses. There's a lot of cool microbe stuff in it.
All right, so I will preface this by saying, I moved to Arizona 13 years ago this week, and I just want to thank Stephanie and Joey and everybody here. I've been searching for ways to interface with communities like this for a while, and this has been a really great opportunity, and hopefully an ongoing opportunity to keep talking amongst ourselves. And we all, as researchers, have skills that I want to figure out how can I apply the knowledge that I have to better help out agriculture in Arizona. And so this is, these interfaces are just great to be able to deal with that. So thank you all for the invitation, and thank you for letting me show a little bit about what we do. All right. We're going to also have some keynote to PowerPoint issues. So this will be fun. All right, we're good. So I sent my talk title to Joey. My talk title is Searching for Next Generation Highly Specific Agricultural Antimicrobials. I sent this talk title to Joey and Stephanie when they were filling out the program, and not 5 minutes later, Joey emailed me back and said, all right, you go to these soil conferences and there's always talks about how to kill things and how to inoculate biologicals. Don't talk about that, talk about something else. I'm just going to steer into the skid on this and then use that as a jumping off point to show what we're doing, and then kind of talk about what we're doing as steps advanced from those like very major topics that Joe mentioned. So one of the first challenges with manipulating microbial communities within agriculture is that we are really good at killing microbes. We have a lot of antimicrobials. We have a lot of things like the chlorine-based treatments. They are very good at killing microbes. They kill all microbes, though. So they kill the good microbes, they kill the bad microbes, they kill kind of everything out there. And so as a next step, we want to move forward to figure out how can we more selectively target the bad guys and maybe enhance the good guys that are out there versus kind of just killing everything. So that's one part of my research program, is trying to figure out how can we be more nuanced in who we target, how can we develop new compounds that are more selective and actually who they can target, and maybe we can even use these as things like prophylactic treatments to prevent E. coli from coming into the leafy greens in Yuma. The other thing, and this is not necessarily a Yuma problem, this is more of an East Coast problem right now, but for bacterial diseases like citrus greening, I was kind of stunned to learn this, but we're cleared for something like 80 tons of streptomycin that we can just spray in the canopies of trees. That's insane to me. Not because for any other reason that, when we start using clinical antimicrobials in agriculture, sometimes it works, sometimes it doesn't. For citrus greening, it doesn't really actually work that much, so we're just kind of spraying antibiotics out there for no reason in a lot of cases. But the other thing that this does is this breeds and gives the microbes a chance to actually evolve resistance to these antimicrobials. If we do that, potentially these resistance mechanisms can bleed over into the more clinical settings where we might end up with antibiotic resistance in hospitals to more clinical antimicrobials that, for a variety of reasons, might be selected for in agriculture. So we want to get away from these broad spectrum, kill everything antimicrobials, and try to develop more nuanced ways to manipulate the communities. Second thing Joey said, y'all hear a lot about inoculants. A lot about biologicals that apparently people are coming and trying to sell their new things to you every week, and there's a ton of these products in the market. I just pulled a couple from the Google image search. How do we make these better? What are the limitations to these? A lot of the ways that these inoculants are developed are somebody's going to go out there and they're going to start culturing these microbes. Maybe it has some phenotypic benefit under certain conditions and certain crops. Under certain parts of the country, and then they market this product across everything to every grower everywhere because it works, in some cases. As a next step, what we're thinking about is how can we develop more specialized inoculants, not just necessarily, here's one microbe that works in one one crop in one area. But can we come to Yuma? Can we learn from the environmental conditions of Yuma? Can we identify microbes that grow here and maybe turn those into inoculates? Hopefully, and I'm kind of the research is kind of, we're still learning a lot about this, but I think there's a lot more specialization in terms of the microbiology out there in the soils. Even if you have the same strains that are, if you sequence them, they look very similar. The strains are going to be specialized to Yuma, strains might be specialized to Arizona. Can we take advantage of that specialization to develop better inoculants for this particular area versus other ones that might be developed for New York or North Carolina or wherever they're coming from before? A side note, a lot of these inoculants, I can tell you this because I sequence things too. There's a lot of inoculants that are basically the same thing. And so, they'll say something like, this is a proprietary mix. I can tell you, if there's a biological, I can probably tell you, 5 of the strains that are in there, and they're probably very similar across a lot of these inoculants in many cases too. So let's develop some more specialized inoculants that might be beneficial for this very area versus ones that are developed for other places. And then the last thing, this is a particular problem to Yuma. Soft fusarium mold of lettuce as a big problem mentioned amongst the growers in the previous workshops. And just talking to people, it's a huge problem out here as well. Can we develop new antimicrobials that target fusarium of lettuce in a way, kind of using those, harvesting microbes from the area to specifically target fusarium? And fungi particularly, we tend to lump microbes together in these broad communities. It's much easier to develop antimicrobials against bacteria than fungi for a variety of reasons. So fungi produce their own certain challenges with that. And I'll talk a little bit about what we're trying to do for that. So what does my lab do? What do we do? What are we good at? Alex and Duke and a variety of other people here have shown these large scale, we can harvest a bunch of data, get a lot of data and try to make sense of that data in a variety of different ways. I'm much more reductionist in what I do. I like growing microbes. I like sequencing microbes. I like engineering microbes. I am terrible at growing plants somehow, but I'm pretty good at growing microbes in a lot of different cases. So what we try to do is actually do a much more reductionist approach and identify characteristics of certain microbes that are good at either targeting bad microbes or helping good microbes to actually develop these communities that we want going forward. All right, so the one thing I'm going to talk about today, and there's a bunch of different things I could talk about in terms of the next generation antimicrobials. The one I'm going to talk about today are called bacteriocins. And so bacteriocins are basically like the broad spectrum antibiotics that you would, if you went to the hospital, you'd be given a broad spectrum antibiotic. Bacteriocins are much more selective antibiotics. They're more selective for a very specific reason. So bacteriocins are produced by bacteria. It's going to be produced and then only target certain cells that have the certain receptors for these antimicrobials. So this circular bacteriocin is going to target the potential target cell with the circular receptor. It's going to not touch the one with the star-shaped receptor. And so the specificity of these happens because we can specifically target these antimicrobials to strains of interest versus these broad spectrum kill everything ones that have been developed in a variety of other ways. Bacteria are very good at killing each other. And so what we've done as a lab is try to just grow and characterize a variety of different ways that bacteria can kill either closely related or more distantly related from other bacteria, and then look at their genomes, try to figure out the molecules that are involved, and then try to figure out can we use these as these next generation antimicrobials. All right, so I'm going to try to keep this at like a 20,000 foot level kind of a thing. One picture, a lot of data behind this picture, but basically the plants on the left have been treated with these bacteriocins to prevent a phytopathogen. The ones on the right are not. Hopefully you can tell the ones on the left are alive, the ones on the right are dead. At some level, in the lab, under very controlled conditions, we've been able to show that these new, highly specific antimicrobials can work to prevent certain phytopathogens from infecting plants. I literally dip these plants in like a soup of bacteria. This works for some phytopathogens, but we also have certain selective antimicrobials that work against some salmonella strains, and only those salmonella strains. We have one antimicrobial that I'm still trying to characterize that seems to work specifically against E. coli 0157. It actually targets the 0157 stands for O antigen. The antimicrobial targets the O antigen, so the 0157 is what makes that strains sensitive to the antimicrobial. So we're trying to develop these highly specific antimicrobials as a way to kind of pinpoint, knock out the bad guys versus anything else in those microbial communities. And then the next thing we want to do with this is not just, throw out the antimicrobial and see what happens and hopefully kill the bad guys. One thing that we try to do also is anticipate what happens if resistance develops. And so if resistance develops to these antimicrobials, can we use that as a way to predict the next treatment? Can we develop these smart treatments that maybe play off of each other? So if resistance develops to antimicrobial 1, can we have an antimicrobial 2 that then is sensitized, the bacteria are sensitized to that? So can we develop cocktails of treatments where they work together in concert to not only kill the first line of cells, but also anticipate the resistance evolving and then kill that resistant cell as it arrives as well. This is just a hole in the hole there means that they're being killed. Lack of a hole means they're not being killed. And so we've been able to do this for a variety of different compounds where we can actually almost anticipate the resistance and then tailor the treatments to try to work effectively. We're doing this in agriculture. You'll see this happen in, they're doing this for like cancer treatments and stuff too. There's a lot of, we're putting a lot of research towards trying to figure out what is the best way to use these compounds in ways that are effective as cocktails versus just singly, like they've been done a lot in the past. All right, last thing I want to mention, this kind of brings in the inoculants and the fusarium side of things. So we're good at killing bacteria. We're starting to learn how to be good at killing fungi as well in the lab. So I got a little bit of money from YCEDA. This started a project this last fall where we are specifically trying to cultivate and then identify microbes that can kill fusarium of lettuce. We're doing this by culturing microbes out of actually Matt McGuire's soil. So thank you, Matt, for sending us the box of soil. So over the last fall, we set up the pipeline now. I've got about 5 undergraduate researchers that are working through cultivating a bunch of microbes, cultivating a bunch of bacteria. This is our first, it took a while to get the pipeline set up. So we have about 100 strains now, hopefully a few thousand by the end of the spring. These are just petri dishes like Matt said. And then what we do, and this was my Christmas break, we started setting up inoculations of fusarium. There's fusarium growing in the middle there, just the bacteria on the left and right sides there. So what we're doing is we're cultivating these microbes from the Yuma soil, and then I'm trying to identify, can any of these microbes actually kill fusarium? Can they limit fusarium in some way? With the hopes that potentially, if they are from Yuma, maybe they're more adapted to the soils here. Maybe they're more adapted to being able to thrive in the communities under the heat, under the salinity, everything else. We haven't found one yet. That's a negative result. The fusarium is kind of thriving on that plate. But nonetheless, we're going to hopefully screen through a bunch of these going through the spring. And then we'll see what happens with that. All right, so with that, again, a ton of people working on this, but special thanks to Stephanie and Matt McGuire for helping us out with the fusarium work going forward, and happy to take questions as well.
Hi, yeah, well, I'm from Yuma area, so I'm interested on this. Do you think this can reduce, I'm assuming every farmer wants to farm efficiently and consume less resources and keep up with the demand. So do you think, like, I'm an irrigator, how can I was saying, we used chlorine and to minimize E. coli and these bad bacteria, right? And you farming this, it can affect and less the E. coli. So how do you apply this on the plant? Like, how can you apply this on the plant? Like, how can I work with this? And where do you see it? What, next year, do you?
Yeah, so this is the exact reason that I wanted to talk to everybody and why this conversation was great. I'm coming at from a very basic foundation with a directed question, right? How can I selectively kill E. coli 0157H7 versus anything else? We got to that point. We're trying to figure out how to scale up and the best ways to do that. And there's a variety of ways. These are natural products, and so we can figure out how to get the microbes to produce a lot of them. It can be an additive. Depending on federal regulations and whatnot, these are also pretty easily moved into plant genomes as well. So we could, GMOs are one thing, but we could imagine one day engineering these kinds of molecules into lettuce, where maybe the lettuce is inherently resistant to E. coli because it has those defense systems already. So again, this is basic foundation. I haven't. We've got some thoughts as to how to scale up and to how to turn this into a usable system pretty quickly. We just haven't gotten there yet. We're still kind of characterizing it at a basic level.
Okay, we've got a question up front here.
Thank you. Great data and very interesting. This is just a thought, maybe not so much a question, but based on Alex's presentation of roots having two different subspecies grow better together, and the fact that soil microbes grow better together, not just isolate, diversity is key. Do you think that when you're researching how to fight fusarium in petri dishes, combining microbes that maybe can kill better together than on their own in different species? Maybe there's research in the soil that those specific species are found with maybe less fusarium pressure and out in the environment?
Yeah, so it's a great question and great comments as well. It's a spectrum of how to do this, right? There's the one versus the community-based stuff, and where we're really lacking is that intermediate. We can get a lot of data and then see trends, or we can do things in a very reductionist way and see things that might not apply. What we're trying to do is build up this community where we have a bunch of different strains, a bunch of different potential inoculants that we can then ask, if we do combine these together, if we do build them into community, is that a better production than just the one versus one or anything else? So can we build a multi-strain community that does this eventually? Again, and then build in understanding of how these microbes thrive under the soil conditions or certain environmental conditions, and then maybe build that into the community as well. We can learn from the large-scale sequencing as to how to make these microbes actually play well together and work as a group together. And as we're gaining all of this knowledge, hopefully we come out with a product that's more diverse than one versus one, but isn't just kind of the data black box that it can be with some of these other studies that are out there. That, again, very useful, just kind of hard to interpret sometimes because it's just a ton of data without the process behind it.
Okay, one more question. We have to do.
Thank you for your presentation. I love the idea of giving a vaccination to a field of plants, maybe if they have an adjacent land food safety hazard. But could you talk a little bit more about how these preventative things are being applied in your laboratory? Are they being applied foliar? Are they root? Are they delivered by chemogations?
Yeah, so how are they being applied? So the stuff we've done so far has been, we just spray them on. They can be applied to the roots in the same way. Other labs have done that a little bit. And then we've actually developed, what we're trying to do now is it's one thing to have the compounds, it's another to have microbes that actually produce these in a way that you not can control, but that you can inoculate to. So basically, can we make microbes that make these compounds that naturally will produce them under the conditions that we want them to produce them under, which I think is probably the best. If we can't make a GMO plant, that's probably the best way to do it, to actually get them out there where we're not just having to apply a bunch of sprays on plants. We can actually have the inoculants out there that might be built into things that y'all are already using to grow the plants. So we're trying to get there, but right now we've just done it. Everything we've done so far is just spraying on leaves as the plants are growing, and then going from there.
Alrighty, good afternoon, everyone. I really appreciate the opportunity to come and speak with you all today about some of the research efforts we've been conducting at Uarizona and West Center. in our efforts to take action and advance method development to investigate the use of microbial activity as an indicator of biological soil health. Just a brief outline of what I'll be talking about here today, just addressing some of the issues of concern which have been outlined previously, but just readdressing them for the purposes of this talk. Speaking a little bit about soil health and what are biological soil health characteristics and how they differ from physical and chemical characteristics, as well as getting into a little bit about some of the current soil health research being conducted at West Center. And in conclusion, just some thoughts as we think forward into the future. So once again, some of the critical issues that are facing desert agriculture that have already been described include soil health, in particular the resiliency of our soils here in the desert Southwest, as well as water scarcity issues. Interestingly, in terms of the U.S. Soil Health Institute, which has put forth a listing of what they term as the ideal indicators of soil health, although growers, I'm sure you all have your own ideas of what those are comprised of. A good majority of those soil health indicators tend to be physical and chemical. The biological indicators tend to be less studied compared to the physical and chemical indicators. Therefore, we need to develop more methods so that we can increase acceptance and usage on a broader scale for those biological soil health indicators as reliable barometers of that health. As has been discussed previously, we've evolved in our thinking over the decades, going from soil quality, which was earlier on described principally as can soils produce crops effectively, and about 20 years post-tense, thinking a little bit more about soil quality with regards to broader environmental quality, animal and human health, as well as enhanced crop productivity. But as we came into the 90s, the term soil health gained more usage and traction. But even still, it was about productive agriculture, which is good. We need to eat, we need good productive agriculture. But we started thinking more about the role of soil macrobiota and microbiota in that process. But now today, here's the term holistic once again. It comes up again and again. And that's because we need that broader based understanding of soil as this vital and living system. And this is because, as we know, and it's already been stated, 1 teaspoon of soil can have upwards of 10 billion active microorganisms. And why are these microorganisms so important? It's because they're driving those nutrient cycles in soil, the cycling of carbon, nitrogen, phosphorus, and sulfur, those critical elements for plants. Therefore, it would seem logical that assessments of soil health should more directly incorporate measurements of microbiological activity and function. However, if we look at the Tier 1 indicators put forth by the U.S. Soil Health Institute, which is actually a recently founded institution as of 2016, and they published the final draft of these indicators in 2018. In terms of what the Soil Health Institute has put forth, these are indicators that effectively demonstrate soil health, and these are indicators that can be demonstrated both regionally and nationwide among various soil groupings. However, if we look at these indicators, we see that a majority of them are physical and chemical, and these are all really important characteristics of healthy soils. We want soils with ideal bulk density and aggregate stability. These are important for a healthy soil, as well as cation exchange capacity and total nitrogen and phosphorus. Okay, no one's going to argue these. However, when we look at the biological indicators, top of the list, crop yield, of course. A healthy soil is going to yield substantially healthy crops as well. Organic carbon, of course. However, nitrogen and carbon mineralization are the only two indicators that are directly indicative of microbiological activity in soil. It would seem that with microbes being such a substantial part, there might be more present. In addition, the Soil Health Institute also put forth a listing of Tier 2 indicators, and they term these as Tier 2 because they know that these require more research. And if we look here, we see a much more substantial listing of biological indicators, some of which have been talked about today. For example, active carbon, we have the fraction of carbon in soil, but what portion of that is actually readily assimilable by the microorganisms present in the soil. And then a lot of the omics technologies, which have already been spoken of today. However, at West Center, we're about applied research. So how can we take the research we're doing and put it to good use out in the community? So our goal at West was to develop a useful biological indicator method that could be performed rapidly, cheaply, and with moderate training that's required. Therefore, for some of the funding that we applied for and received from USDA and NIFA, one of our goals was to develop a rapid biological indicator index to assess soil health utilizing ATP technology. Now, ATP technology has been studied quite a bit. However, it's kind of ebbed and flowed in the scientific realm. This is due in large part to method issues, how to best extract ATP from cells, plus the 2-week preconditioning requirement of soils, which is just not practical nor feasible. As many of us remember from middle school or perhaps even high school, not microbiology, but just biology, ATP is the energy carrying currency of all living cells. It's a really critical molecule. And these 3 phosphate groups, as present in 3, can really indicate the energy levels of a cell. Okay? And so we can have 3 phosphates for ATP, 2 for ADP, and 1 for AMP. And the more phosphates that are present, the higher the energy level of a given cell. We did a lot of method development work with a company, because at West it's a public-private partnership center. And we did work with a company out of Canada called Luminultra to evaluate their kit for use with granular material to assess ATP levels. Once we developed our methodology, we wanted to then be able to assess the influence of wetting drying cycles or water stress on the metabolic activity of soil microorganisms. And this company we worked with, they were really excited about that because they hadn't done any work with soils, and they supported us quite a bit in these efforts. To this end, we evaluated 5 moisture-stressed Arizona aridosols that we obtained from Campus Ag Farm. They were a combination of Pima soils, Gila soils, and Brazito. And they had not received any rainfall in about 60 days, so they were definitely moisture stressed. We utilized the data that we obtained in calculating and assaying for these 3 different species of AXP, so the ATP, ADP, and AMP, to compute an AMP stress index. And this is essentially just the ratio of adenosine monophosphate, so that lower energy form, to ATP, which is the higher energy form. Given the results that we obtained, just a quick idea of the type of information that we were able to garner about our samples from these results were that samples that were characterized by high levels of the higher energy form, or ATP, and low levels of AMP were characterized by higher rates of active microbial metabolism in soil. Therefore, greater numbers of active microorganisms were present, and the stress index that was computed was lower, so lower levels of AMP. However, for the soils where we saw low levels of ATP, so lower levels of that high energy molecule that were present, and high levels of AMP, were typified by lower levels of active microbial metabolism in soil, greater numbers of potentially active and dormant microorganisms, and therefore a higher stress index value. What do these results tell us, at least preliminarily? Well, one was that the AMP stress index values are sensitive to moisture content changes during these wetting drying cycles. And these are really critical, and we obtain these results in near real time. So just a really quick assessment of what we did. The soils were under moisture stress at a moisture content ranging from about 2 to 3%. We adjusted them to at or near fill capacity, about 13 to 17%, depending on the soil type, and then allowed them to dry under 30 degrees Celsius back down to where the moisture content levels were back to pre-amendment levels. These are the corresponding microbial activities that were measured under these different moisture regimes. So under the low soil moisture content levels, where the soils were under these moisture stresses, the microorganism communities were subjected to extreme microbial metabolic stress and potentially even dormancy. And we were calculating high AMP stress indices, so those stress index values were very high. During periods where the moisture content was ample and ideal for the communities, little to no metabolic stress was observed as we were measuring low AMP levels. However, upon drying, once again, we saw this return to extreme microbial metabolic stress and dormancy. Fortunately for us, and what we'd really like to see in our work, is that this stress index, these trends were similar across the 5 different aridosol soil types, which varied by texture, salt levels, and total organic carbon. And this is really critical for method development work because we want to be able to apply this to as many different soil types as possible. We don't want to just be able to apply it to one. We want to be able to use it across different types of aridosols here in our area. What are some of the outcomes and implications for growers here? Well, now there's this method that we've been working on and have developed. We would really like to be able to field test it that hopefully can be utilized to rapidly assess metabolic activity levels of microorganisms in soils in near real time. Based on how the kit works, the method we were able to develop, ATP levels can be measured in less than 5 minutes, ATP and AMP for computation of the AMP stress index, less than 15 minutes, and all 3 species to compute an adenylate energy charge, less than 25 minutes. This is ideal. It's a lot less tedious than the methodologies that have been previously published. This is vital information for growers because we know that a metabolically active soil community is going to decompose organic matter, thereby generating inorganic nutrients that are critical for plant growth. And they're going to continue to serve as those critical drivers of nutrient cycling in soils. Therefore, it seems that a metabolically active soil community can be an effective indicator of soil health, and is. However, further research is needed. These are preliminary studies conducted with microcosms in the lab. We need to be able to vet this technology at field scale, but we also want to be able to combine it with some of the other technologies that are available to assess biological soil health, including the omics techniques that were described previously, and most importantly, crop yield. So is the data that we're getting, can it eventually be correlated or serve as a predictor for crop yields? Secondly, in terms of the microbial monitoring that we did and would want to implement for agricultural soils, could this method be employed to help further optimize timing, frequency, and duration of watering or irrigation events? We conducted a 7-day wetting drying study or moisture stress study with those same aridosols. It was a 7-day study once again, and we calculated the stress indices at multiple time points. On the left-hand side of the table are the time points. Middle column are the stress index levels that we calculated, and on the right was the interpretations. And once again, what we saw was prior to moisture amendment, really high stress indices that dropped readily upon the addition of moisture to at or near fill capacity. However, by day 4, this is after 4 days of incubation at 30 degrees centigrade, we started to see a turn in our AMP stress indices towards moderate metabolic stress. So we started to see our microbial communities coming into a stressful type of environment. And by day 7, those stress index levels had returned to pre-moisture amendment levels. So what are the impacts of this type of data, and what could it have for growers? Well, it can provide an enhanced understanding of microbial activity trends in desert soils. So how are our microbes in our soils behaving given different moisture regimes that we're experiencing here in the desert, just as part of the normal climate here? Also, it can help us better prepare for water resource scarcity, which we know is an issue that we face here. So if we know that water cuts are coming, we can have an understanding of how our microbes are going to react to less water. And then lastly, as we convert more desert land to arable land, we can apply a sustainable conservation-based approach. Ideally, my future aim as a researcher is to help develop a practical toolkit of biological indicators that would be most useful to growers in arid regions, such as the desert Southwest, to help ensure soil health and resiliency. Thank you all.
Thank you. I really enjoyed the presentation. It was cool. I was curious with the stress index. Since it's a ratio, is it possible you could have a good ratio, like a low AMP stress index, even though you have low overall microbial activity? And if so, do you envision like pairing this with characterization of that through a different indicator?
Yes, so, of course, it's all preliminary work, right? And we haven't really even tapped to the usefulness of this assay. I think in particular where we started was these moisture stress regimes and looking at responses accordingly. But I do think that there is likely to be, I'm sure there is actually, a suite of indicators that would pair really beautifully with this type of data. Because in and of itself, what does it tell us? What would it tell a grower? Okay, we have lots of ATP, that's great. But what would be ideal to do is to develop a suite of indicators that growers can utilize out in the field. And this is a field portable technology, by the way. I didn't mention that. But that's our thinking, is to have a technology that can be utilized by growers to provide good information in consort with other factors and indicators. I'm just not sure what those are yet.
Hi, sorry if this is a silly question. I'm an undergrad student again, so when I hear some of the things that everyone's talking about, and I think about the research you're doing, and you're talking about adding water and microbes, and then earlier speakers were talking about the actual water that they're adding to the fields, which have been treated, chlorinated, potentially killing microbes. Like, when you do research like that, do you use the same sort of treated water that farmers would be using, or would you be using DI water? Or, I mean, is it apples and oranges?
I really like that question because another aspect of my work is antimicrobials. And so that was an avenue of work we were interested in going down. We'd have to do some assay modification for that because we'd want to make sure we differentiate any background ATP that's there in soil. So some cool half-life studies would be in order for that. With ATP that's associated with viable living active cells. But I think that would be a really good question, but we'd have to modify our assay so that we are definitively detecting what's live versus what's been killed in the soil. But I am really interested in that, and it's great that you're an undergrad here asking questions. I love undergrads, so yeah.
I'll speak loudly. So this is great. Is there any way that you convince the board of directors of the Central Arizona Project and all the senior staff at the Urban Arizona Department of Water Resources that their preferred method for stealing our water is like seasonal fallowing, which means we're going to kill our soil every summer, and all the other research you guys do gets thrown away, and we try it again when the winter vegetables start, and then we kill it again in the summer? Can you convince them? You've got about a month. You can take this.
Yeah, actually, I really appreciate that because it was stated earlier, like what happens to the fields in the summer and how can we maintain soil health in the summertime. And I think this is a promising technology that could generate a lot of data and information to help convey those issues of concern, I believe, and to work towards efforts to maintain soil health in the summer months, and hopefully convince them of those arguments in particular. So I'm on board. Thank you.